The first step of the cell cycle is interphase. This is the nuclear envelope. And this is a centriole. And right here is the chromatin. During interphase, the first part is G1, where the cell increases in size and the organelles duplicate. The next part is the S phase, where DNA replication occurs. First, the DNA unravels. Next, an enzyme called helicase separates the bases, resulting in a replication fork. Then the DNA polymerase matches the bases with their complementary base in the 5' to 3' direction. On the leading strand, the polymerase constantly goes in the 5' to 3' direction, while the lagging strand goes from the 5' to 3' direction in fragments, and it is slower than the leading strand. Lastly, ligase connects the bases back together with hydrogen bonds. These DNA strands are semi-conservative. The very last part of interphase is G2, where the cell expands. All of these phases above are checkpoints to ensure the cell is not damaged. Now onto mitosis. The first part is prophase. Spindle fibers are created thanks to the centrioles. These are chromosomes, which are a form of DNA. During this phase, the nuclear membrane vanishes. This is a chromosome. A chromosome is a DNA molecule made up of two sister chromatids connected by a centromere. The next part of mitosis is metaphase. During this phase, the chromosomes move to the middle and the centrioles move away from them. The following step is anaphase, during which the chromosomes, which are also called sister chromatids, are separated by the spindle fibers. The blue sketches represent the individual chromosomes. Now to telophase. Notice how the shape is no longer circular. This is because it is preparing for cytokinesis, where the cytoplasm splits. Telophase is when the nuclear membrane starts to reappear. Now we start cytokinesis. During cytokinesis, the cytoplasm splits, and two different cells are produced, resulting in 2 brand new cells. In an animal cell, it is split by a cleavage furrow. For a plant cell, it is split by a cell plate. The parent cell is the original cell. And the daughter cells are the result of the cell cycle. Even though they all have different names, all 3 of them are identical. The cell cycle is extremely important because without it, cells would not be able to sustain these things along with many others. And on top of this, wounds would not be able to be healed.
Thanks for watching!